when you kind of integrate everything together with quality understanding, you can really make a big difference on several fronts, whether it comes to making new drugs, avoiding side effects, and reducing the cost of healthcare. These are the big questions that we face. And Dr. Butte is really at the center of it. I don't actually think it's an exaggeration to say that he is among the leading lights, if not the leading light, in this field called translational informatics. Translational informatics is this field where we think not just about one area, but how to translate from one area to another. So it's this idea of translating from biology to the clinic, or translating from economics to the clinic, that sort of stuff. And this is really the area where he has really made a mark, and why we're really so fortunate to have him here today. Now, he's this big shot, but I will tell you that I have been deeply impressed by the time he's given me. I first met him when I was a PhD student, and it wasn't even face-to-face. It was over the phone, and he talked to me like about an hour on the phone when I was just a PhD student, and he was a professor at Stanford. And over the years, I've met him at conferences. He's always made time for me, and it's not really because I'm that amazing. I look around and see he does this for lots of people. One of the things that's really unique about Atul is that he's really not just about his enterprise. He really cares about the success of our field as a whole, and particularly the health of all of us younger people that are coming behind him. And I have deep, deep respect for him in that regard. I think in a lot of ways, even though I've never been a PhD student or postdoc or junior faculty in his department, he has really become one of my academic mentors, and I'm deeply thankful to him about that. I'll tell you that our interactions are really interesting because they follow a familiar pattern. I come to him with a very small-minded question that's normally concerned about my career or how I get a grant funded. And then, Atul usually reminds me that there's bigger things in the room. There's bigger things in this world, and he wants to change the world. And I remember, actually, yeah, I do want to get tenure, yeah, I do want to get that grant funded. But really what we're doing here in science is really, truly taking our best shot at changing the world. So with that, if you could just really join me in welcoming Dr. Butte.
Great, so thanks for having me here. Obviously, I've actually never been on this campus. I've always, when I've given grand rounds and stuff, been on the medical school side, so it's kind of a real pleasure to be over here. It's a beautiful campus. I can see why undergrads like this place now. So I'm a medical doctor. Usually when medical doctors start, we have to start with our conflicts of interest. I have just a few. Suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You probably don't want to believe another word I say over the next 50 minutes or so. I wouldn't blame you. But I'm most proud of the bottom right. Those are all the companies started by my grad students. More than half my grad students start companies now, even if they go into academia. And they do this with the most amazing platform in the world, and that's data. It's often big data, and it's often open big data. So I'm going to show you how they do it. I'm going to show you how I do it, and maybe I'll convince you this is the most amazing time to be in biomedical research today. I don't think it's taboo at all to talk about companies. You're going to hear companies throughout this talk, and I don't care. I mean, I think it's just part of the innovation process now, as you can see. In my lab, we do have a saying that if you want to change the world, you can't just keep writing papers about it. You actually have to file IP. If no one licenses it, it's up to you to start the company, get it to patients. So we do this a lot. So a lot of people use this as a slide of shame. This is my slide of significance. So I'll start with that. If you haven't heard, we're in the middle of this data deluge. This is my favorite example of that still. This cover of Economist magazine from 2011. This guy's collecting ones and zeros with the umbrella. And this is the issue where they announce that the human species is generating 2 zettabytes of data every year. If you forget your metric prefixes, it goes kilo, mega, giga, terabyte, petabyte, exabyte, zettabyte. And of course, next year it's 4 zettabytes of data, right? Because this number's doubling every 2 or 3 years. And I'll be the first to admit most of these zettabytes of data are these YouTube videos of kittens, right? Playing on pianos and yarn, cute stuff. Entertainment value, but no scientific value. But there's scientific data in the zettabytes as well. So for example, the Large Hadron Collider generated petabytes of data to help those scientists find new subatomic particles. My favorite example is NASA. Okay, so NASA announced about a year and a half ago, by the end of this decade, with the new observatories and all, they're going to be taking so many pictures of the sky that they're thinking they're going to generate an exabyte of data every day. Okay, NASA, an exabyte every day. Now that's crazy. That's a huge number of hard drives. But they already admit, though, they generate so many pictures today There are not enough astronomers on this planet to look at all the pictures. Okay, it's a fact. So what do you do when you have too much scientific data and not enough scientists? You have to get new types of scientists to help you. So they literally crowdsource this. So you can go to Galaxy Zoo, you can take a test, and you can assign, is this a star, is this a galaxy? NASA gets citizen scientists to help them analyze this data because there's no other hope, basically. But that's the lesson you're going to keep hearing, that when you have too much scientific data, at a certain point, you cannot just depend on traditional scientists to look at all this data. It has to be opened up to a new community. And of course, that's what we're going towards. Now, in fact, there's so much data that this guy, Fritz Anderson, who was the editor-in-chief of Wired. Now, Wired is not the most scientific of journals, I admit. But in 2008, he wrote, that there's so much data, science itself is getting obsolete. Now, how do you say science is getting obsolete? That's kind of ridiculous, right? Actually, it's a scientific method that is getting obsolete. Okay, so here's the thinking, right? What is the scientific method? We're all taught that in second or third grade. We start with a question, or we call it a hypothesis, and then we go make measurements to answer the question, right? We did this in grade school, everyone does this, right? You measure the plants or whatever to test your question. But we're now in the world where we already have the measurements. 99% of the hard part is figuring out what's the question I want to ask. It's really the hard part, right? What's that killer question that I can ask and answer now with this data that everyone's wanted to know the answer to this, and no one's realized we can actually ask it right now with this data. That is the hard part. It's not writing code, it's not the databases. I mean, those are things you gotta learn. But figure out what is the unmet need, what is the killer question is still the hard part. And that kind of reverses the scientific method. In my lab, we're kind of agnostic to diseases. You're gonna see, we don't really care which disease we study, but we go after the ones that we have enough data that we can actually study, right? Maybe we don't have enough this year for one, we'll skip it for next year, right? We choose the questions based on the data, but that's the hard part here. And of course, we're there in this kind of molecular world as well in biomedicine. A lot of ways to show data delusions in biomedicine. I love to start with these affymetrix gene checks. I could have put a sequencer, I could have put a mass spec, proteomics, metabolomics, I could have picked any of those. But I got my start, I guess 18 years ago, analyzing these gene checks. And now they're so cheap, I carry one in every suit. So I have one in my hand here, right? Big data comes from little packages, just like this. And you can put a sample of cancer or a normal tissue or animal model, and this is a readout of every gene in the genome. This happens to be the RNA, I'm showing on this chip. But it's kind of amazing. I mean, basically everyone uses these, a couple $100. But you know what? When you get tired of measuring them one by one, we actually now measure them in the 96-well format. And that's so cheap, I carry that in my bag here. That's what's kind of falling apart here. You can see what they look like over there. That's the 96-well format. And you know what? When we get tired of measuring them in the 96-well format, we now have a 384-well format. And that's so cheap, I carry that in my bag. I cannot more clearly illustrate exponential growth in biomedical data than to show you these plates. And we don't even use these anymore. We've moved on to RNA-seq. And this company doesn't even exist anymore. They just got acquired 6 months ago, Affymetrix. This is how fast the world is moving. And everyone runs these chips, right? It's not even a big deal to run these. What happened in this field is that so many people start to run these gene chips, they start to write papers. Here's a list of genes, and there's a list of genes. Here's a list of genes of heart failure or in cancer. And they started submitting papers. And what happened is it started with the journals. The journal said, hold on a second. We're getting too many of these papers. This is around 2000, 2001. We're getting too many of these lists of genes. No more papers with these gene chips. No more. Unless you deposit this data into an international repository where other people can double check the math. Probably getting important to do that, getting closer to the clinic. And maybe others can use these samples for something else. Okay, so the precedent for this idea of sharing data actually goes way back before these chips. It goes back to something called GenBank. GenBank. It's a next door neighbor to PubMed if you think about the tabs up there. So GenBank is like 38 years old now. Way older than the World Wide Web. Way older than like 3-inch discs, if anyone remembers those. Older than CD-ROM TVs. I mean, they were sending tapes to each other because the first paper came out with the DNA sequence. Nobody wanted to type in AGTC again, right? So you got to send this stuff to each other. So precedent is set that if enough scientists use a molecular modality, you got to share the data openly. So fast forward in this field to August of 2012, when this article came out in Nature, featured my lab and many others, we hit a milestone in August of 2012, 1 million publicly available microarrays, 1 million of them. You see the growth rate from 0, right? So I started my career, probably like right around here, and we went to 1 million. Now, if you look really carefully here, kind of looks sad, right? Doesn't it look like it's slowing down there at the end? These are two sites, Array Express is the European one, Geos and NIH one. Kind of looks like it's slowing down there. Can you see that? Then you realize they only counted half the year, right? Still doubling every 3 to 4 years. We're just a few thousand shy now of 2 million. Okay? If you look right now, these repositories were like 1,980,000. Maybe by the end of this month, we'll hit 2 million samples publicly available. Now, most of these are human samples from some disease studied over the past decade and a half, just sitting there. What does that mean? You can get lost in the millions and billions, so let's make it concrete, right? Any cardiologist can go to this website and start to download cardiac samples, whether mouse models, anyone on my campus can download cancer samples. Whatever you want to study, probably there's some data to get started with. Actually, forget about our scientists. Even a high school kid, right? A high school kid who needs to do a science fair project. Even she can go to these websites. Let's say you go to NCBI Geo, search for breast cancer, hit search, and find and download nearly 60,000 samples of breast cancer. About as easily as she could find a song on iTunes today. The vast majority human, there's mouse models, rat models, dog models, synthetic constructs, cell lines, biopsies. What's so magical about 60,000 samples, you might be wondering? Let me tell you, that's more samples of breast cancer that this kid has now than any breast cancer researcher in this field. Because every one of them eventually has to submit data into the middle, right? And whoever figures out the middle database now has more data than any one researcher in the field, right? It's a no-brainer. Maybe you don't like breast cancer, it's colon cancer, prostate cancer. Maybe you like diabetes. Probably someone has studied your disease of interest, especially the common ones, and the data is already sitting there publicly available. Now, actually, the number I like here is 2445. Let's go like 2400. 2,400 research labs, research groups essentially have generated data to submit data into this repository. 2,400 labs. So let me tell you what a ridiculous number that is. Let's pretend for a second. Okay, let's pretend I'm a newbie researcher in breast cancer. I know nothing. And I'm writing an NIH grant, and you're all my NIH study section reviewing my grant. And I propose to you in my grant that somehow, as a newbie researcher, I'm going to get 2,400 of the best breast cancer labs in the world to take their best characterized patients to run the latest technologies, and I propose to you they're all going to give me that data for free, okay? You would laugh me out of the room if I proposed this to you in a forward direction. Yet here it is. 2,400 labs are there to help you as a newbie for free. So when I think of public big data like this, I actually think of it as retroactive crowdsourcing. What does that mean? 2,400 labs are there to help you. They've been helping you, and they didn't even know they were helping you, right? Actually, 2,400 labs are there to help you, and you don't even know they're helping you. And for some reason, 60,000 samples aren't enough for you. Just wait another year or two, it'll be 100,000 samples, right? In this hour, we'll add another 150 or 200 samples, just sitting here, right? Because that's how many people are generating data. And in general, nobody looks at this data. It's just sitting there, waiting for you. All of this data is just sitting there, waiting for you. Now, if you have any doubts a high school kid can do it, here's the high school kid that did it, okay? So this is Brittany Wenger. I've still never met her. She was in Sarasota, Florida. She's a Duke undergrad. She's probably a grad student now. And at 17 years of age, she won the top Google Science Fair competition, first prize, for building an artificial brain neural network to diagnose breast cancer. She downloaded immunohistochemistry data, some qPCR data. She built a classifier, and she won top prize for building a diagnostic. I guess she was bored, because the following year she was in the newspaper all over again for doing this with leukemia. And of course, what I mean is if a high school kid can do this, we all better learn how to do this now. I don't care what field you're in, we all better learn how to do this in biomedicine. In California, actually, we're going crazy now over this kind of thing, right? You know that we're all trying to teach all of our kids how to write code, right? Starting at age 7 now, we're trying to teach every kid at age 7 and beyond how to write code. And because of that, we're dropping cursive. Remember, we're learning cursive? Not required anymore in California. You better learn how to do Python coding instead of cursive. Much more useful skill. So, but these are the kids that are coming right behind you. Okay, so we've got this middle gap of people who don't know how to write code. They better learn in a hurry here. Okay, because these are kids that are coming here. This is the new way. I'm not talking about replacing what biology here. I'm just talking about starting with data first instead of hypothetically, right? Start with figuring out what people have actually done first. So that's what high school kids do. And now, where are we going with medicine? We're talking about precision medicine. Now, President Obama's got us all thinking about precision medicine. It's been a year and a couple months when he mentioned the State of the Union address. And obviously, there's a lot of money going forth. Even today, the grants are getting reviewed, actually, as we speak, for these big coordinating centers. A lot of this is data-driven, but precision medicine actually makes physicians really nervous. I can tell you, it makes physicians really nervous. Why is that? Because actually, if we're moving into this era of precision medicine, then by definition, the last thing we were doing wasn't really precision medicine. It wasn't precise at all, right? So doctors don't like even being told that, right? Like, that's not precise what you were doing. So they kind of argue with you. And actually, you start to think about, like, what is a precision of a medicine? How do you say if a medicine is precise or not? So the way I think about precision medicine is actually to think about how we classify diseases, right? All those little pigeonholes we can put patients into, like how do we classify a disease? The word for a classification of disease is called nosology. It has nothing to do with your nose, okay? Nosology is a systematic classification of diseases. It actually goes back to this guy, Linnaeus. Linnaeus is the guy who put all the species into a taxonomical tree, remember? Kingdom, phylum, class, order, family, genus, species, remember that? We all had mnemonics. Some were clean, some were dirty. Don't get them here. You all had them on a test, okay? Kingdom, phylum, class. Well, Linnaeus is actually the first guy to put diseases into a tree too, right? It's called the General Form in the 1700s. You can see how they try to make a tree with the fonts here. That's the best they can do in the printing presses. Now, he's not remembered for this because he got it completely wrong, okay? He's got fevers over here, fevers with pain over there, mental problems over here. He got the species so correct. Remember the poor soul for that. Why remember for this? But he launched 200 years. The thing to do, if you were a learned philosopher or physician, was to make your own classification of diseases. Leading up to 1860s, William Farr and the Royal College of Physicians came up with what we now call the International Classification of Diseases, or the ICD system. It wasn't called that back then. We're now in ICD-10. We just moved from ICD-9. We'll talk about that in a moment. But the thing to do is now that is how we classify diseases. And actually, it's lost all connection to science. It's actually just making sure we get paid the right amount, right? We've lost the scientist in this at all. But I started to think about diseases and disease classifications. Here's ICD-2, the second edition. So it's interesting to study how we classified disease 100 years ago. And actually, I have this book in my hand here, right? When old doctors die, this is their trash. You can buy these like on these old book sites. It's like $30. It's like gold to me. Second revision, Paris, 1909. So this predates even the United Nations. This eventually became a League of Nations Day, so if you know your European history. And this book is amazing, okay? It's written the same way our code books are written today. It says, okay, use code 39 for cancer of the umbilical cavity, that's a cheek. Cancer of the stomach, liver. Use code 41 for cancer of the intestines, cancer of the skin. And then this book does exactly what we still do today, 100 years later. We're running out of numbers, put all the other cancers in 45, okay? Including lung cancer and brain cancer. Not worth distinguishing from each other. 800 years ago, I kid you not, it's in the book, right? Actually, you can learn a lot about medicine from what we put in not otherwise specified. It's all the stuff we can't diagnose, we can't treat, can't do anything about, essentially. You learn a lot about what the unmet need is by looking in NOS, actually. Lung cancer is the #1 cancer killer, I think, today. Was cancer not otherwise specified 100 years ago. And literally, though, this book literally does, it has to say, I've even flagged the page here if you're really curious. This book has to say that if you run into a patient that died because of a visitation from God, be sure to use code 189, But the same way you're laughing at this 100 years later, they're going to be laughing at us in 20 years, right? Or 10 years. Those silly fools didn't know this bowel disease and this joint disease were the same thing, right? They're gonna, because we're still classifying diseases based on the kind of doctor, right? Instead of actual molecular change. So the visual to think about is we're going to replace this with this, right? We've got to get to the molecules here, right? Now, we were making steady progress. I don't doubt that. This is ICD-2. We've had ICD-9 for 30 years. Last year, we were finally replaced with ICD-10, the 10th edition. And believe it or not, the physicians are kind of up in arms about it because there are all these random codes. You need more codes. But there's even a code, believe it or not, if you were surfing and hit by a whale, there's actually a code for that, right? But you have to have a code for everything. A lot of people were making fun of these codes. A lot of memes all over the internet. My favorite is this one. Yeah. I don't always get sucked into a jet engine, but when I do, I use ICD-10 code 397.336. There's even a code for getting sucked into a jet engine. Now, I actually have a lot of friends in San Diego. Okay, so I give this talk, and there's a big naval base in San Diego. And my friends there have told me, indeed, you can get sucked into a jet engine if you're on an aircraft carrier, right? But this is the chronic form of the disease. Okay? So I can imagine getting sucked in once, but not twice. Okay? But there's a code for it. Okay, there's a code. Actually, need that, right? So this is like, just in case it happens twice, there's chronic. So, but this is fun. We can make fun of this, right? I don't doubt that. But what we should have been doing is making fun of ICD-9. Okay, so let's go back to lung cancer for a second. ICD-9, 162.9. So these are all the things that fall in 162.9. So do we take into account the latest in gene sequencing like EGFR sequencing? No, of course not. Do we take into account molecular testing for subtypes? No, of course not. Do we even take into account the microscope, where we can tell large cell from squamous cell from adeno? No. Do you know how we used to classify lung cancer in ICD-9? Left lung or right lung? Okay? If you ever need a definition of imprecise medicine, here it is. And ICD-10 doesn't even fix all of this, and we should stop fighting it, and we should get to a mode where we as physicians and scientists communicate with each other in the right way what we think our patients have, and more importantly, tell the computers what we think this patient has, so the computers can start to help us, because we're gonna need that help as we move forward here. And we shouldn't be laughing at ICD-10, it doesn't even fix all these problems here. That to me is precision medicine. So step one though in precision medicine is to figure out better diagnostics. How are we gonna tell what patients have which diseases here? And so I think a lot about diagnostics, and in fact, my first big NIH grant was all about these microarrays. And we had a whopping 25,000 when I wrote this grant, now we got 2 million. I said, you know what, I think we should just go get every human disease studied by microarrays, just go get them all. So we started an effort to just collect every human disease in the repository studied by microarrays. I would collect disease and healthy, disease and healthy. I'm not going to show you genes, but then every gene is a box here, and the colors mean they're more or less. We can calculate these signatures. Marina did a lot of the early work in my lab. She made this slide. And so we, people have calculated signatures forever. But remember, I can take any one disease, and I got dozens, hundreds, thousands of people studying these diseases. So I can do what's called a meta-analysis here, right? I don't have to trust any one data set. I can figure out what's in common across all of them. And now just for aficionados for a second, now it's kind of always dangerous to show a central dogma, but I guess we're in a kind of undergrad. Maybe we should show some biology here for a second. This is central dogma here. DNA codes for RNA, codes for protein. I love protein diagnostics, okay? I know you can make big companies with RNA ones like Genomic Health. I love protein diagnostics. It reminds me of my days as a pediatrician long ago. It's still amazing to me that you can have a toddler screaming with a fever and sore throat, and somehow you stick this Q-tip there, and then you drop some drops on it, and there's enough antigen that it'll pick up strep, right? You can tell the strep test. That test is called an ELISA. It's like 1970s proteomics, okay? Enzyme-linked immunosorbent assay. I love ELISAs. But you can't do an ELISA for the entire proteome. You got to pick and choose, but they're pretty sensitive. And you work in a desert. You don't even need a refrigerator. So I love protein tests like that. And we're measuring RNA with these chips. And we got a whole bunch of these. So if the RNA is up in the disease, maybe the protein is up because of central level. Kind of simple, stupid thinking logic here. So we did a bunch of these. I'm not going to talk anything about these. So we had one paper out on cancer markers. To make a long story short, medulloblastoma was a pediatric brain tumor, and kind of nasty. The surgeons were saying, we can't really tell what's the cancer, what's the normal brain, so they wanted to paint. They wanted to paint the operative field, turn off the lights, have it glow, and maybe you can tell what's a cancer, what's not a cancer, right? So how the hell do you design a paint like that? Well, you got public data on medulloblastoma, figure out which genes are up, figure out of those which are cell surface proteins, figure out what's not in astrocytes, glial, and neural cells. You can digitally subtract all that. And you got some candidates, and you just stain, and you can see these are brighter, these are darker in medulloblastoma than the normal cerebellum. So we kind of came up with some things like that. We came up with transplant rejection markers. When patients get a new organ put in, some days the patient wakes up, they don't feel this, it's not painful, but the organ, the body says, this isn't part of me, I gotta get rid of this thing. That's called acute rejection. You can't tell it's happening, so we routinely stick in long needles to see if it's happening. We wanted a universal blood test for rejection. We didn't care which organ got transplanted. If it is being rejected, we wanted the same test done. So here's an example of kidney, liver, and heart transplant, all upregulating the same marker that we could detect in the serum as well. So that got published. The one I'll spend a little bit of time on is talking about this with preeclampsia. Have any of you heard of this disease, preeclampsia? Show of hands. Yeah, so it's usually about a third of the audience. It's a third of the audience that watches Downton Abbey because one of the characters dies in this disease. Spoiler alert, sorry if you haven't watched it, right? That's usually the only reason Americans have heard of this. You'll see usually that's when you're pregnant, that's eclampsia, preeclampsia. It's still how husbands lose wives around the world. It's a nasty disease, billions of dollars. And this disease falls right between the cracks. So what do I mean? I love my friends obstetricians, but they say the delivery is the cure. Just take the baby out, the mother's better in 48 hours with some mag sulfate. And then we pediatricians have to keep these tiny babies alive for months, right? So it falls right in the cracks there. It's hard to, the pediatricians can't say anything about keeping the baby in because the mom's might even die or seize or something. And so there's 4 drugs in trials right now for preeclampsia, if you look at clinicaltrials.com. But we're still using the same diagnostic from 50 years ago, looking if there's protein in the urine. You know, in today's obese America, that's the most non-specific thing to go on. So we said we need a more specific test for preeclampsia, okay? So Linda, who was a grad student, started work. Bruce did proteomics, and Matt got involved later. If you just search the repositories for preterm birth, preeclampsia, 266 micro-experiments already done. I don't mean 266 chips. I mean 266 collections of chips, right? Here are 299, 249, 154 samples, 143, 104. You get to pick and choose whatever ones you want. You know, pick the best ones you want or the biggest experiments. Informatics methodology, I can just simply imagine a big Venn diagram, right? I don't trust any one of these experiments, but I trust what they get in common, right? What's everyone seeing upregulated in terms of RNA? You know, just see what's in the intersection of the best ones. And you chase those down in protein form, and here's an example of serum protein, hemopexin that's up in preeclampsia versus normal. We tested this with samples, UC Stanford samples, we've got some samples from repositories, and you can see this example that's up. So we've got Marchidon's funding to start. I got one of these CTSA grants to get going, a seed grant, and we were able to test the samples. Now, what do you do next in Silicon Valley? You start a company on this. So you can see a couple of these arrows here, okay? So you always start with the unmet need. We don't write a line of code unless you know why we're doing this. What's the question? Public data available, Marchidon's available to get it started, data analyzed, design a diagnostic, a seed grant from our CTSA, $50,000, launch a company, $2 million seed financing. Now, let me be crystal clear to this academic audience. I'm not showing you $2 million to brag about. Actually, it's not a lot of money to raise for a company. I am showing you $2 million because I'm convinced now this is the new way science will continue out of academia. We've gotten the grant. We've written the papers. If you want to really change the world for these patients, you got to get it to an actual diagnostic. So you file the IB, you start a company on this. And the science continues in a startup company. It's a multi-center prospective trial that has to launch you, right? Now funded on $2 million of private money. Or to put it another way, do you know how hard it is to get a brand new $2 million NIH grant today? You know how hard that is. The science continues in a startup company. And there it goes. And actually, this became a one-man company, Matt Cooper, okay, who just started to really figure out all the teams he could work with. I love this story because we already sold the company. It's already acquired. Some of you know what that means, right? So let me just illustrate the timeline involved. Public data, started public data, which people think is as valueless as kitten videos. And we designed this diagnostic. We test samples that are in shelves and repositories. We get the papers published, we launch the company, get the seed financing, sold the company. Start to that finish, 24 months. Company's already launched, already acquired. Inventors happy, investors happy. Can we do more of these? Of course we're gonna do more of these, and I'm giving away my secrets here, okay? Because every one of you could pick a different disease in medicine, and we would never overlap. Such is the need for diagnostics in medicine. So this is just our diagnostic story. It's a big one, right? This is how we're just going to keep doing this. But this is how fast you can actually get these companies launched today. The science continues in a company. Let's talk about therapeutics. I think therapeutics are much more of a mess. I think the rate limiting step to precision medicine is going to be therapeutics. We're going to have 5 ways to classify a cancer and have the same drug for all 5, because we're not going to have anything different going for people, right? This is the rate limiting step. Now, if you ask lay Americans, how much does it cost to develop a drug? It's amazing, lay Americans know anything in common, but they know this number, $1 billion, 10 years, right? Because it's advertised on the Super Bowl. I mean, this is how you kind of justify pricing, I think. $1 billion to develop a drug. But actually, if you do the math, actually $1 billion is an underestimate. How do you get the real number? It's really simple math, okay? Here's the formula. How much pharma R&D did you spend? Divide by the number of drugs you got, okay? How much you spend, divide by the number of drugs. So Matthew Herper does this every year in Forbes. If you look at the top 12 companies, they spend between $4 billion and $12 billion per drug. Kind of not sustainable anymore. We cannot keep counting on the pharma and biotech industry to come up with these drugs. Let me put it a different way, okay? You may love the pharma industry, you may hate the pharma industry, I'm not gonna pass judgment on it, right? But let me put it a different way. Even if the pharma and biotech industry was 100% successful in everything they do, there are not enough of them on the planet to get us all the drugs we still need. And then you realize it's gonna come down to each one of us to solve this problem, okay? Look, I have a lot of friends in the biotech industry. I kind of consult for them all. You saw my second slide, right? But I still don't have faith they're gonna solve this problem. It's gonna have to be us to solve it, even in academia. There's no choice. So how are we gonna do it? I mean, we're gonna try our take at this. And so we figured out, can we start to use public data to try to solve this problem, to get to some more therapeutics? So while we're collecting disease and healthy samples, people start to put their drug experiments out there on the internet. So I start with the Whitehead Institute, Broad Institute, Todd Golub, and Justin Lamb, the connectivity map. Now it's called links. But they take cells treated with drug, without drug, with drug, without drug. So with drug, without drug. On the left, we got with disease, without disease. On the right, we got with drug, without drug. They're not even the same kind of cells, but we're going to put these two together. And the reporters for this call this Match.com for drugs. If you know Match.com, you know, it's kind of how you find a date, maybe a mate or a spouse. And there's a famous saying, opposites attract, okay? So I'm going to show you all the informatics with my arms. If I've got a disease where this gene goes up and this gene goes down, and I can find a drug that can make this one go down and this one go up, maybe there's a match there. That's 2 genes, imagine 20,000 genes. I don't have to talk about Pearson correlation coefficients or Kolmogorov-Smirnov tests, you get the idea here, right? And so I'm just naively looking for drugs that reverse the kind of molecular pathology I'm seeing in the disease. It's kind of that simple. There are 2 C words I'm not using here. The first C word I'm not using is cause. I don't think the drugs are getting to the cause of the disease. Okay, I'm never saying they do. Actually, medicine, I don't think we have any drugs to get to the cause of the disease. Maybe statins, that's about it. The other C word I'm not using is cure. I'm not saying these drugs are a cure for this disease. It's just a treatment, okay? Kind of naive, that's the approach. Turn our crank, get lots of ideas. Here's a chemical, there's a chemical. Where we get a lot of good ideas is not the new chemicals, but the new uses for the old drugs, drug repositioning. We didn't invent this concept of finding new uses for drugs. There's some classic examples everyone should know about. The first is an interesting cardiac drug that had a side effect of hair growth, that is minoxidil today, right? That started as a cardiac drug. But of course, everyone knows the other cardiac drug that had its own interesting side effect. Patients don't want to return that drug after the trial. Of course, that's Viagra, right? That started as an angina drug. Fewer people know that Viagra has done a double switch. There's another drug called Verbatio, same molecule, but specifically indicated for pediatric pulmonary hypertension. So the blood vessels from the heart to the lungs, they get constricted, kind of opens those blood vessels up too. People were teasing babies on Viagra and all that. But you know what? Instead of finding these new uses by accident, how about we find them on purpose using public big data? That's the concept here. Turn the crank. The computer's humming, and we got lots of ideas. There's a drug, there's a drug. And how do you test these? Well, I don't know. You got to run a clinical trial. Oh my God! Before you put this into patients, you got to test this in animal models, right? I mean, you might love or hate animal models. You got to test them in a preclinical model. And all my animal model friends start to scatter, okay? Because they know I'm going to create a lot of work for them. We had a good set of collaborators. I mean, great folks that really did a lot of work with us. But today now, when I really need to test a drug in an animal model, I go to the other thing that changed my life. AssayDepot.com. Kind of sounds like Home Depot. And you would be right. Okay, let's go browsing. Biology, chemistry, DMPK stands for drug metabolism pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology, pharmacology and toxicology. Okay, let's click on pharmacology. That seems safer. Okay, what kind of mouse do you want to run today? Bone, cardiovascular, dermatology, diabetes, urinary infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. I guess they say I'm a diabetes guy, so let's click on that. Here, I can order an OB diabetes model, 16 mice. Maybe I have 2 groups of 8, with or without drug, or I need to test like 4 groups of 4, maybe a dose ranging study. I'm going to test my mice this way, right? If you don't know what OB is, it says, this mouse has been eating a lot and getting diabetes since 1951. Okay, this is the standard model of American diabetes. Okay, it's not the best, it's not the worst, it's one that everyone uses. And in 28 days, they'll measure the fasting blood sugar, the insulin tolerance, stress, glucose tolerance. That's all the stuff kind of do in humans, they're doing the mice. Now, what am I covering up here? The price, $9,000 for this service, 9-week turnaround time, and literally, I kid you not, add to shopping cart. I can buy an entire translational mouse model like this over the internet with a credit card today. What the hell is this, you're asking yourself, right? These are called contract research organizations, CROs. They've been around forever. Pharma uses them. But because of Asking Depot, Science Exchange, there are a bunch of these now, they hawk their wares on these kind of websites. Pricing transparency, prices plummeted, that even we in academia can afford this now, okay? It's unbelievable. Now we still use collaborators, but now this is not the hard part. Now some of you run mice experiments, I know that. And you look at this, 16 mice for $9,000, that is ridiculously expensive, okay? And I admit, that's because you have slaves. I mean, graduate students who could do this for much cheaper, right? I mean, God. But for dry bench scientists like me, what does MasterCard say? This is priceless, right? For me to show a mouse picture of my computational paper, man, this is priceless, right? Now, some of you with really sharp eyes notice this is run in China, right? Some of you may have a problem with that, others not. I'm not going to say I don't have a problem with that. But let's just go further here. When you actually hit add to shopping cart, you're presented with the 133 companies that actually will do this experiment for you for a price, including Maryland and Wisconsin. So just because it's outsourcing doesn't mean offshoring. You can pick wherever the hell you want this thing done in the world, okay? Maybe you're worried about quality. I'm sure you are. Well, you just click on certifications here. Click here, FDA, there's 5 of them. You need them, ISO 9001, there's 7, AAA like 28. You click on the certifications, it's only going to show you the companies that have been certified that way. Oh, how I wish I could find the right collaborator on my campus with the right certifications. So it's not some random undergrad doing the experiment. I wish I could get to that level on my own campus. You can't. Yeah. Now, some of you know. Some of you are super concerned here. All that computer stuff is whatever. Based on this, you're going to humans next. So you're still worried about the quality of this data, right? I know you are. Usually at this point, people are really unbelievably concerned. What the hell am I doing here, right? I'll simply say, if I really, really, really need to know if this drug is going to work in this animal model, I just ordered 2 of them from 2 companies. Hell, at $9,000 each, I can order 10 of them from 10 companies. Now, why am I saying it like this, right? Think about a mouse researcher who gets a mouse experiment to work in their lab. Did they go across the hall to get a completely separate team of researchers to repeat that experiment? Nobody does that, okay? We're in a so-called reproducibility crisis, they say, right? Nobody does that, but I can hire as many independent teams as I want if I have the money, right? So what I've done suddenly is I've taken that quality problem in your head, I've now made it a function of dollars. You want better quality, just get more dollars. Don't have enough dollars, maybe you're going to get 1 or 2 of these to work. So we typically will pick one from the West Coast, one from the East Coast. Nothing against flyover states, I still love you guys. We pick one from these 2 coasts, and we just get these experiments done. And literally, this is how we get these things, because we don't trust. So here's kind of weird thing to think about. I don't trust any one of these public data sets, but I trust what I get in common. I don't trust any one of these companies, but I trust what I get in common. And it's a new way to kind of surf on top of the data and the validation to get to findings in a much more efficient way. By the way, I don't have to love these mice anymore, right? So I can work on diabetes one week and cancer the next week, right? I don't have a mouse colony. Right? It's all just by money. So that's a kind of advantage here. So we don't know about a dozen of these. This is my favorite example. We got on the front page of Wall Street Journal for this one. My famous rat colonoscopy videos. Nobody's laughing, so let's illustrate the geometry involved. This is a colonoscope. This is a rat. There's a company that knows how to do this. Why would you ever want to learn how to do this? This is a normal rat colon. It's anesthetized, but not, you don't have to sacrifice. You can see this is a normal pink, healthy colon. Rat with Crohn's disease. It's a chemical called TNBS, makes skin inflammation there. Rat all better with topiramate. The seizure drug topiramate, we predicted works on Crohn's disease here. Okay? That's the seizure drug topiramate. So 40 megs of video. So by the way, nobody at Stanford knew how to do a rat colonoscopy. So this one I was still at Stanford. So what it means is I'm not just outsourcing to get to save money. I'm outsourcing to get the best damn data in the world. It's a company that specializes in this, right? We've done a dozen of these. I'm not going to show you all of them. This is still my favorite. The psychiatric drug is antidepressant called imipramine. We predicted works on small cell lung cancer. Okay, small cell lung cancer. Humans smoke to get small cell lung cancer. 5% survival rate. This mouse, if you knock out P23, RBP 130, this mouse gets mouse lung cancer. So this is not a PDX, a xenograft. This is a genetic engineered mouse file. You can see the nodules there. So these are good friends. Julian, Alejandro, Nadine in their lab did the work. Joel was in my lab who made these predictions. Now this mouse is all better with imipramine. So what's imipramine? It's a tricyclic antidepressant. We don't use it anymore. It's still in a pharmacy. We replaced it with serotonin reuptake inhibitors like Prozac. Imipramine had some side effects. In some people, it makes you really sleepy. And in others, it might set you up for QT prolongation, might set you up for arrhythmia. Actually, neither of those two side effects are as bad as lung cancer, 5% survival. And cancer's melted away. It's gone in this mouse. Why do I show you this one of the dozen we've done now, right? So we show the public data, which people think is valuable. So we make the prediction. We test cell lines in a dish. I'm not going to show you that. We get this amazing mouse picture. 15 months after the computational prediction, we got IRB approval. We launched a clinical trial on this one. Patients dosed on this one. Total cost of the trial, $50,000. CTSA grant, okay? Not $1 million, not even $100,000. Because the drug's already there, you just pay for physician time and some CT studies, right? Now, I'm financially connected to this drug, so I'm not part of the trial, just to be clear, right? And this trial, actually, believe it or not, of those two side effects, I actually never thought sleepiness was the worst of the two. So we're going to still have side effect issues in repositioning as well. Why sleepiness? Because if you've been told you only have 5 months to live, you don't want to spend on sleeping, okay? I grant you that. So it's actually a problem. So we've got to look for other tricyclics. You know, as they say, search for keys around the lamppost to figure out ones that don't have that. I don't care if this trial fails. We need to do more of these. Nobody's working on targeted therapeutic right now for small cell lung cancer, right? We have to go after way more of these and launch many, many more of these in academia. So I don't care if the next 10 of these fail for me, okay? I'm going to do way more of these. Here's one that's not published yet. Okay, so this is still coming out. So this is Peter, Martin, and Mazen was in my lab doing this work. This is a disease called psoriasis. I think you heard psoriasis, thickening of the skin. The keratinocytes go crazy. This is a genetic engineered model, it's an overexpression model that has psoriasis. Now, I'm neither a psoriasis person nor a mouse person, right? This mouse kind of looks scruffy, you can't see the fingers. Okay, the digits look clear here. This is after the drug. By the way, what is this drug? So this is a 1960s diuretic, okay? This one we don't use anymore. We have way better diuretics, okay? 1960s diuretic, whether topical or systemic, this mouse gets better. Actually, let me show you the cooler picture. That's a cell picture here, okay? So here's wild type. This is what normal mouse skin looks like. This, the pink are the keratinocytes. Overexpression concentration, which actually mimics human psoriasis, keratinocytes go crazy. They go deep. You can see them growing along this hair follicle, right? So they go deep. This is half the dose. This is double the dose. You can see the keratinocytes are all back to where they're supposed to be. That's kind of cool. But the most amazing part of this is actually the blue. Now let me explain this for a second. If you ask any biotech company, how do you treat psoriasis? They'll say, oh yeah, we've solved psoriasis. We've got these monoclonal antibodies. They kind of wipe out that part of the immune system, but yeah, they treat psoriasis too. It's like hitting with a sledgehammer, right? And that's only $50,000 a year for those monoclonal antibodies here, right? The blue here are all the immune cells. This is the first small molecule that actually fixes the keratinocytes and is not an immunosuppressant. Nobody has a molecule like this. And it was sitting there in public data, a 1960s diuretic. And so many more just sitting there, waiting for the right use. Right? There's no molecule on the planet that does this right now. So I'm not just talking about minor little uses like curing cancer. I'm actually thinking about fixing the biology too here. Okay? I mean, I think there's so much more sitting there waiting for you because nobody else is doing this. And what do you do in Silicon Valley? You got to start a company, right? Because then all of a sudden it becomes a business decision. So Numeti is now launched more than $5 million. You always start with unmet need, data available, NIH funding, Arrow money. StartX is one of our incubators. We have incubators on both Stanford and UCSF campuses now. The incubators are great to launch companies. Think about them, launch them. And believe it or not, Numeti is working with Allergan. So until 2 weeks ago, Allergan was merged with Pfizer to be the biggest pharmaceutical company in the world. They're not merging now. But this little team of 7 full-time people is developing the next psoriasis drug for Allergan, the behemoth Allergan. So I call these garage biotechs. For some reason, we revere garages in Silicon Valley. They're national monuments. HP and Apple started in garages. So I think the next Genentech, the next Amgen is going to start in a garage. I don't mean making meth in your garage, no. I mean cancer drugs and these, right? That's what I mean, right? I think this could be done. So you got lots of company. So in the last few minutes, let me just talk about where we're going next. What's the next big open data that's happening? What half my lab is invested in? If I need to skate to where the hockey puck is going? Clinical trials data. What's a clinical trial? The most expensive experiment in the world. And half of them fail. And we've got this weird culture that when they fail, we don't even write papers about, right? It's got to change. And I don't mean that summary table they're making you put on clinical trials like, oh, I mean the raw clinical trials data. How to reanalyze, how to do meta-analyses, meta-studies. EMEA, which is the European FDA, is requiring raw clinical trials data release. FDA might get there. They work with IOM and NAS, and there's a lot of acronyms here, but there's a lot of fights. I think we're going to get to say where raw clinical trials data gets released. I run for NIAID, Allergy Infectious Disease, this thing called import. We give out, we're the only repository where we literally give out the raw clinical trials data for 100 trials. I mean, each encounter, each patient, each drug, each arm, de-identified, of course, but this is the new world. We just had some papers out recently on how we reanalyze clinical trials data. I'm a big fan of reanalyzing this, and go sign up, and it's a free account, you'll be approved in minutes, and you can go download like crazy all this clinical trials data, because really we're the only ones that you download it, you run it on your own computer. You don't have to play on someone else's computer or anything like that. And all the others are just having trouble like that. Let's get non-molecular for a second. How many of you in this audience have a wearable or Fitbit? It's usually a good, yeah, okay, so it's about 5% of the audience. Usually it's 30% Silicon Valley, but on the East Coast it's like nothing. Nobody still gets into the East Coast. Now I used to laugh at these people, quantified selfers, right? They measure everything that comes in and out of the body. Measuring things that go into your body sounds tractable. Measuring things that come out of your body sounds gross to me, right? Why don't you voluntarily do that? In the hospital ICU, we call that ins and outs. That's such a chore for the nurses. Why would you do this at home, right? So I used to laugh at these people until December of 2012. I came back from a trip to Hawaii. I stepped on a scale. I was 247 pounds. Then I realized when I hit 250, they can measure me in tons. That's a quarter of a metric ton. It's like, what the hell happened to me? I'm driving everywhere. I'm not walking. Bought everything Fitbit makes. Lost 50 pounds in the last 2.5 years. I carry a scale everywhere, including in that bag. Every morning I weigh myself, compensate for what I eat based on that. I'm a pediatrician. I know what we tell the kids with anorexia not to do this. I still do it myself. I weigh myself every morning because I need to know, right? That's how I compensate. And so it's funny. I lost my 50 pounds. All my friends on Facebook know I lost my 50 pounds because it's a very social thing, right? It's probably the most important social intervention. It's not for everyone. I'm an early adopter. I admit. It's called the Hawthorne effect. You just measure things, they get better. It's also Weight Watchers rules. You bite it, you write it. So I've recorded everything I've eaten in the last 2 years. It's kind of interesting. That's all fine. And my friends all know this. But when I go to my doctor, and it's been written in the newspaper, Sutter Health, which is neither Stanford nor UCS, the competition of both, it's kind of embarrassing. But my doctor, and it's been written in the newspaper, they spent $1 billion on Epic. Okay? $1 billion. But my doctor was the last to know I lost the weight. Because their $1 billion system doesn't talk to my $40 gadget. Can you smell the opportunities here, guys? Okay? Can you see what needs to happen? If we don't get with the program, other companies are going to come in and take the healthy patients away from us. Okay? It is already going to happen. I know it's only a few hands now, but it starts with the watches, it starts with the wearables, and people, healthy people, have more rewarding interactions from the companies than they're getting from our own clinicians. And just saying Epic doesn't support it is not an excuse anymore, okay? Even if Epic doesn't support it, we all have to get with this program to connect our patients between encounters. There's so much more data, I don't even have time to talk about microbiomes, ENCODE, Cancer Genome Atlas, all this other kind of data that's out there. So a couple things I'm really proud of. In California, we tend to stay ahead of the curve. So one of the things that we launched with state money is a precision medicine initiative. Our governor launched a whole year before President Obama. It's only $3 million for the first year. We're upping it to $10 million hopefully this year. And we're giving out pilot grants. I'm lucky I get to work with Governor Brown and the state of Sacramento to try to steward this money to give out to interesting pilot projects. So it's not, it's important to convince your own state to get involved because there are a lot of companies, a lot of jobs that are created this way as well. So I'm proud to be part of that. I'm running now this Institute for Computational Health Sciences. We're slumming it out in this brand new building until they build our brand new building, opening in 2019. Amazingly positioned between Illumina and the brand new hospital, Women, Children, and Cancer Hospital. As the real estate agents say, location, location, location. You couldn't buy a better lot for computational folks like us between Illumina and the hospital, right? It's kind of interesting. And so the goal is to build the strongest team in the world in biomedical data analysis, improve our education, got to recruit at least a dozen faculty. We're hiring postdocs and students recruiting students like crazy, organize infrastructure, and build and develop our new data assets. And that's what I'm going to end with is our data assets. Why did I leave Stanford after 10 years? I love Stanford. But we actually have something at the UC system called UC Health. So UC Health is this umbrella across not just UCSF, but UCLA, which both of those two are top 10, Irvine, Davis, and San Diego. And if you actually do the math, so I'm in charge now of all the patients across all of those 5 systems. This is a little bit of an old slide, but we're up to 15 million patients get some care in the University of California. So that's 4% of the US population. And I'm in charge of all the records of all of them. So actually, I wanted to build a centralized data repository for all of it. Actually, we've already built it. I wanted this done before my first year anniversary. So we built it. We have this massive Dell server sitting in San Diego Supercomputing Center. If you think HIPAA is bad, FISMA is even worse. So this is other FISMA back, firewalls and stuff like that. And we've kind of built out this database. So to give you an example, we have 1 billion lab tests. Okay, we literally have 1 billion lab test measurements just in this repository from these 15 million patients. What are we going to do? Well, we're incorporating all these 5. I'm not going to spend a lot of time. We're building a clinical data warehouse. We're starting with the clinical data, like in Epic and Allscripts. And we'll get the imaging data, pathology, radiology as we go. There's genomics, but genomics will come a little bit slower unless we get a research kind of collaboration going. We're going to target all sorts of big, money spenders and put a financial use on this. What could we do with this data? These are 10 use cases for the 10 campuses of the University of California. I think clinical researchers can run GWAS across all of the, we're heading towards getting genetics on all 15 million. We're trying to figure out how to do that, but that's definitely on our radar. Community activists might be able to download data, match it up with Cal EPA data. Patients will get a harmonized data set across all of UC Health, app designers maybe. So we're going to slice and dice and de-identify this data, give it out to entrepreneurs, give it out to the people in the state, because we've got to use our data to leverage a lot more than just clinical research, improve operational care. Chief medical officers are desperately looking for ways to predict who's going to get readmitted, within 30 days or 90 days. Cancer genomics researchers. What am I trying to do with this? Is I'm trying to build maps. So these are maps of death and disease, and that's kind of morbid. But think about Google Maps. It shows you how to go. Maybe places to avoid, I guess. Here, here's an example of a map we built with real data, and each circle here is a disease. So patients come in with alcoholism. A year later, a bunch come in with liver cirrhosis. A year later from that, they come in with liver abscesses, or they go directly this way. And the squares mean they've died of these diseases. So this is an example of a map, right, of how Californians go from health to disease to death. You get a little bit more complicated. So here patients come with a heart attack. They might die right away. A whole bunch end up with heart failure. That means the heart isn't pumping as strong as it could have or should be. Some end up with lung disease. That's a no-brainer. The fluid kind of backs up. And then a whole bunch die of sepsis. I never realized that a patient with a heart attack can die in 3 years from sepsis. I never really connected the dots that way. Or they might go this way. And so we've got these maps of just how do you go from health to disease for Californians. And my plan now is when we open our brand new building in 2019, we're going to have a room full of monitors, the wall of monitors, and we're going to have our maps there. Now, Google Maps is great, but the one thing Google Maps does not do is it actually doesn't show you all the cars, right? It kind of shows you traffic, but it doesn't show you all the cars. And 2019, when I build that new room with a room full of monitors, and we're going to plot the maps, we are literally going to plot all 15 million patients and how they transition from disease to disease to disease to death. This is with real data. This is a prototype right now using death data. But you can see as age goes up, these are Californians and how they're going from disease to disease. A lot get this disease, that sepsis, a lot die from there. And we're going to plot our 14 million patients, and we're going to predict what's going to happen in the next 90 days. And we're going to predict what's going to happen in the next year. And we're going to do something about it. What can we do to prevent the mortality and morbidity we're predicting with machine learning what's going to happen next? And to me, that's going to be the new definition of an accountable care organization. One that actually accounts for the 14 million patients under its care. And not just encounter to encounter, but with the wearables and mobile devices and everything in between. And this is what we're building in the University of California. So to close out, what is big data and biomedicine in the end? It's about cloud computing, algorithms, programmers, Hadoop. I didn't even mention any of those things, but of course it's about all these things. But to me, it's about predicting diseases before they strike, explaining rare diseases that defy experts, finding drugs for diseases that lack attention. There are not enough pharma companies on the planet, for our needs. Making sure we do the right, safe, cost-effective thing for patients. It's an amazing platform for biomedical innovation and entrepreneurship. I sum all that up with one word, hope. I think this has to be the future. We cannot depend on the pharma and biotech industry to make these drugs. I think patients are counting on us in academia to do this. It's up to us to step up and figure out how to solve this. It cannot keep taking $1 billion in 10 years to do this. We have to find more efficient ways to get there. But we're going to be the hope in academia to actually get us there. Can't do any of this work without a long list of people helping, 40 plus people across the University of California that are on my payroll, basically helping us get all that data in one place. I would color code them based on site, but actually we are all one University of California now. So I don't tend to make divisions like that. We actually think it's one now. And I have a long list of collaborators, Alejandro Julian with the mouse model, and Jay Prashvika and others with the cancer, with the GI mouse model, and Mike and Tom and a whole bunch of other folks working with us. And I thank UCSF for endowment support. My lab is blessed with 17 NIH grants for these 9 institutes of NIH. These 4 give me more money, these 5 give me less, but I still love them. A bunch of grants, JDR, HPH, HMI, that's been gone. Stem cell money, disease-specific money, seed grants from both UCSF and Stanford. I always thank my admin and tech staff. Mary's my admin. I'd never get a grant or paper out the door without them. I always thank Zach, who's been my friend and mentor for my life. Keith and Sam for recruiting me to UCSF. I always thank my daughter. She's a science nerd and comfortable with that at age 13, and we got to keep more kids in this field. Biology doesn't have to be about dissecting frogs. It's as cool as downloading data now, right? Let's keep biology exciting for them. And I always thank my wife, Tanya Deshpande, a molecular biologist biochemist. She reads every major paper grant that comes out of my lab. She does collaborate with us. She's been a co-author on many papers. She starts companies with me now. Lets me go all over the place to give talks like this. Thank you very much. We're a little bit past the hour. I won't be offended if you run out of the room, but I think I can start take some questions. Yeah, please come this year. And I'll repeat the question if you're still recording. Okay, you have your hand in the back. Yeah.
On the racks theorisis.
Yeah.
I was trying to, and you said you had a diuretic drug. I mean, I was unable to get where is the data. I mean, yep, it was there. I mean, did you use data to find that?
Absolutely, pure data, pure public open data. So another thing I want to make a point out here is, I have no fancy microscopes. I have no fancy mass spec. I admit everything I show seems easy to do, but technically you can do everything I just showed you because you have the same data I do, okay? I don't have any private data, right? Especially with the drug stuff. So literally people have been studying drugs like crazy and their effects on cells. It's something called the connectivity map. It's now called links. So you got 18,000 what they call perturbagens on that side, and you got all these other diseases studied by migraines by other investigators on this side, and we just put these two together. Sometimes out of convenience, maybe there's a new researcher got recruited into our campus, maybe because we care about these diseases, we'll go get the contract services done. But literally all that data on the drug and the disease were publicly available. There's another repository I didn't mention called PubChem. PubChem is an amazing repository. Any of you heard of PubChem? Yeah, a couple of hands go up. So nobody uses data of PubChem. So PubChem are these screening assays, right? If you get a cell line to glow, colorimetric assay, if you get it to glow like when it's living or dying, if a receptor is hit, once you get it to have a colorimetric assay, then you can put it in front of robots, and robots can test 1,000 or 1,000,000 drugs, right? One by one. So those are called drug screens. The pharma industry does a bunch. And NIH started really funding them back in 2001. This is an NIH roadmap initiative. But if NIH funded you to do this, you got to deposit that data in something called PubChem, okay, all that screening data. So fast forward today, what's in PubChem now? Think of it as a big grid, okay, big matrix. 140 million compounds have been tested in 1.1 million assays or screens, okay? So if you do that math, it's about 170 trillion little boxes, of which 8 billion of them have a measurement of this chemical in this screen, okay? I could easily bet you a beer this beats any pharmaceutical company. I could actually bet you a bottle of wine This beats all pharma companies combined, because this is doubling every 2 to 3 years. And nothing in pharma discovery is doubling every 2 to 3 years, right? We all know that, right? And they give you this data without a user ID and password, right? Probably every future drug is a column in this grid, and it's just sitting there in PubChem, right? So how do you even know about these repositories? How do I even discover them? The best catalog right now, we don't really have good catalogs. The best catalog is something called Nucleic Acid Research. It's a very obscure journal. But every January issue is called the database issue, okay, where they just happen to take on this role. And the database issue basically has papers on all of these databases, like PubChem and all of those, right? So at last count, I think last January, it was something, I think it was 1,700 papers in that issue. Okay, you can't even print this thing anymore. It's got to be online, right? 1,700 papers. And just to give you a sense of scale, all of NIH databases were 2 papers of the 1,700. Okay, that's how many public databases are out there right now. Not only is every database growing exponentially, the number of databases is growing exponentially, which is a double exponent, which is actually still an exponent if you do math. That's how crazy this open data field is right now. And nobody uses this data. Other questions? Thoughts? Sure.
Thank you for a very inspiring talk. So in the beginning, you kind of go from, we have this very clinical classification system.
Yeah.
We dive into molecular etiology, but in the end, I guess with UC Health, going back to
the ICD-9 codes.
Exactly. So do you foresee a future where we just have super intelligent AI just do everything based on molecular etiology, or you foresee a different future and how we perceive?
No. So everything we're doing has machine learning and AI and all this, right? But remember my third slide, the hardest problem today is figuring out the question, okay? Look, we could throw unsupervised learning at this stuff. You got all sorts of patterns everyone's already known forever, okay? To get to something interesting does take a long time, okay? So you tend to go up to supervised machine learning. You have a question, you're trying to get an answer to that. And so, but figure out the cool question there is the hard part, right? Computational people have a hard time learning what to do with all this data. They have the methodology, they got the toolkits and all this. So, but it's hard to figure out what to do with all that. So what I encourage usually computational people to do is go learn what the questions are. They have something amazing at the hospitals called grand rounds, okay? Can you believe every week they get an expert flown in to tell you about a disease, everything that's known, and everything that's still yet to be discovered. And they even feed you breakfast, but it's at 8 in the morning, so it's kind of insanely early. So I tell computational people to go to grand rounds, go to medical seminars and learn what is the latest, right, in this field. That's the hard part, is learning what to do with all that data. So, but it's not just turning on an AI, because you're going to get, I had some computational folks in my lab, and they were looking at lab test data, and they said, you know what, there's this amazing correlation between complete blood counts with differential and complete blood counts without differential. What is that? Why is that so important? Well, actually, it's just an arbitrary thing, These are the simplest, kind of stupidest things that they're finding. They're going to find 10,000 of them before they get to something innovative there, right? So you're better off knowing something about the field and going after it. That's my opinion from the AI perspective. Finding the questions is the hard part, Other questions, comments, thoughts? Yeah, you're with the back of that one. Go ahead.
The questions you're saying, finding the questions. I mean, there may be a million questions out there. How does a computational guy even start?
Yeah. I mean, so it's really about tractable questions. So like what I want my computational folks to really get to. So most of the people in my lab are hybrid people in the end. Because you can actually major in this stuff as an undergrad, right? Then they go to grad school, they go to postdocs. So I like hybrid people, but the more computational minded ones, what I want them to do is I want them to sit in grand rounds. Let's say it's a grand round of pancreatic cancer. I want them to hear from the expert. I want them to realize, I have data on pancreatic cancer in my database right now, I can see they don't have a good therapy. I can see they don't even have a company working on this stuff yet, right? So maybe I can mash up this and this, and here's my expert I can go talk to, see if I get something to work, right? That's the kind of inspiration I want them to have at grand rounds, right? Because you have the methodology, but you got to know what to use it for. Otherwise, it takes forever to get to something interesting, right? So usually diagnostics and therapeutics are really good places to start.
Yeah, so thanks a lot for your time.
Thank you very much for having me.
You guys are able to come, and thank you so much.